An example of a mutation in the patient's gene is indicated by a highlighted base pair in a DNA double helix. The RNA polymerase enzyme transcribes this gene to make a corresponding mRNA containing the same mutation copied from the DNA sequence. This mRNA is translated into protein by the ribosome at the position of the RNA mutation. The ribosome incorporates an amino acid that is not typically present at that position of the protein. As a result, a mutant protein is made. In contrast to the healthy version of this example protein, which is efficiently exported from the endoplasmic reticulum, ER, the mutated protein does not undergo this normal export process and, as a result, accumulates in the ER, leading to the cell becoming sick. Sickness at the cellular level translates to illness at the higher order histological level and at the organ level, leading to observable signs and symptoms of disease pathology in the patient.